Dear students, the next chapter is Molecular Basis of Inheritance Part 1. Let us discuss some important 5 mark questions from this chapter for final PUC exam. Question number 1. List out the salient features of double helical model of DNA. It is for 5 marks. For this, we should write some important valid points like the first point is Watson and Crick. Based on X-ray diffraction data, it is produced by Wilkins and Franklin. They proposed a simple and famous double helical model for the structure of DNA. In this DNA molecule, two polynucleotide chains are present. In each DNA polynucleotide chain, phosphate and sugar acts as a backbone. Along with that, nitrogen bases are present at the lateral side, project inside of the DNA molecule. In the DNA molecule, the both strands are complementary to each other. Along with that, these two DNA strands are present in anti-parallel direction. That means one strand present from 5' to 3' end, another strand present from 3' to 5' end. The very most important point, Chargaff rule. According to Chargaff rule, in between two DNA strands, hydrogen bonds are present in between purines and pyrimidines. In this adenine, it forms 2 hydrogen bonds with thymine only. Cytosine, it forms 3 hydrogen bonds with guanine only according to Chargaff rule. The next point is the DNA 2 strands are coiled in right-handed fashion. At this time, it produces pitches. Each pitch have 3.4 nanometer length. And in this pitch, it consists of 10 base pairs. In each DNA strands between two base pairs, 0.34 nanometer distance is present. At the same time, the DNA it have a width that is equal to 20 angstrom units. Additional hydrogen bonds confers the stability of the helical structure of DNA. So these are the valid points for first question. Second question, explain the transforming principle experiment conducted by F. Griffith. It is also for 5 marks. F. Griffith, he conducted a series of experiments on mice with Streptococcus pneumoniae bacteria. In this Streptococcus pneumoniae, it having two types. One is a pathogenic S strain and non-pathogenic R strain. So S strain bacteria, it causes pneumonia disease because of a mucus polysaccharide coat around it. But this coat is absent in R strain so that it become non-pathogenic and never cause any disease. So with these two strains, F. Griffith, he conducted a series of experiment. In this first step is, so S strain bacteria, it injected into the mice and mice is die due to pneumonia disease. In second step, he injected R strain bacteria into mice and it is alive due to not causing disease. In third step, he taken S strain bacteria and he killed this S strain bacteria with hot water. It is called heat killed strain. This heat killed strain bacteria injected into the mice and mice is live. And finally, he injected both heat killed strain bacteria and live R strain bacteria into the mice and mice is die. After this experiment, Griffith concluded that the live R strain transformed by heat killed strain and it become virulent and cause the disease is called as transformation. But Griffith not explains the chemical nature of transforming material. For this, the three scientists Avery, MacLeod and McCarthy, they purified DNA, RNA and protein from heat killed strain bacteria and they concluded that the DNA's enzyme inhibits transformation and finally they concluded that DNA is the hereditary material in bacteria. So these are the valid points for second question. So when you write these points in this way, you will get full marks. Question number 3. So explain the Hershey and Chase experiment on E. coli bacteria with bacteriophages. It is for 5 marks. So remember this question having both points and diagram. So the valid points for this question, Hershey and Chase, they conducted experiment on E. coli with bacteria infected viruses called bacteriophages. They prepared two types of culture medium with radioactive phosphorus in the form of P32 and radioactive sulfur in the form of S35. So the bacteriophages which grown in P32 medium, it consists of radioactive DNA. At the same time, the bacteriophages which grown in S35 medium, it consists of radioactive protein coat. So the bacteria infected with P32 virus, in this bacteria, radioactive DNA is identified inside of the bacteria cell. The bacteria which is infected with S35 viruses, in this bacteria, radioactive protein coat is identified outside of the bacteria cell. So with this experiment, they confirms that viral DNA only enters into the bacteria host cell during infection, but protein coat of virus not enters and it present outside of the bacteria cell. So these are the valid points for this question, and along with that you should diagram. So it is a two steps experiment in this first step and second step with the 3 stages and 3 steps like infection, blending and centrifugation. So then only you will get full marks. Next question is explain the Mendelssohn and Stahl experiment on E. coli to prove semi-conservative DNA replication. It is for 5 marks. In this question, it carries valid points with diagram. The valid points are, so Mendelssohn and Stahl, they proved the semi-conservative DNA replication in Escherichia coli by using heavy nitrogen in the form of N15H4Cl and light nitrogen in the form of N14H4Cl. So they prepared the two types of culture medium with heavy nitrogen and light nitrogen. The bacteria which grown in N15H4Cl medium, it consists of heavy DNA molecule and it form heavy band after centrifugation with cesium chloride CsCl density gradient technique. And the bacteria transferred to N14 medium for one generation, and after one generation the daughter bacteria cells having DNA, this DNA form hybrid band. So the bacteria after second generation in N14 medium, its DNA forms both hybrid band and light band, and this band it confirms that semi-conservative DNA replication. So after DNA replication, the newly synthesized daughter DNA molecules having one parental strand and one newly synthesized strand, that mechanism of DNA replication is called as semi-conservative DNA replication. These are the valid points for this question. Along with that, you should draw this diagram. So in this diagram, the first step it forms heavy band, and in second step it forms hybrid band. In third step, it forms both light and hybrid bands. Question number 5. So explain the process of semi-conservative DNA replication. It is for 5 marks. The first point for this question, one parental DNA will divides into two daughter DNA molecules is called as DNA replication. And this DNA replication, it is catalyzed by one of the enzyme. It is called DNA dependent DNA polymerase enzyme. It always synthesis the DNA in 5' to 3' direction only. The second point, ORI site. It is also called as origin of replication site. So the DNA replication starts at a specific point is called as origin of DNA replication or ORI site. So DNA polymerase never initiate replication so that some more enzymes are needed for replication process. The two DNA strands are separated by breaking hydrogen bonds to form a replication fork during replication. Deoxyribonucleotides, they act as a substrate and they can provide energy for replication and shows dual function. So the DNA two strands acts as a template strand for replication. In this, the template DNA strand which having 3' to 5' polarity, it replicates continuously. This strand is called as leading strand. And the one of the another template DNA strand which is present from 5' to 3' direction, it replicated discontinuously. It is called lagging strand. So finally, after completion of DNA replication, the DNA fragments are joined by ligase enzyme. So these are the valid points for this question. Along with that, you should draw this diagram. It shows continuously synthesized and discontinuously synthesized strands with different polarities. So then only you will get full marks. Question number 6. Explain the mechanism of transcription. It is for 5 marks. So the transcription means the process of formation of RNA from DNA by RNA polymerase enzyme is called as transcription, and this transcription always occur from 5' to 3' direction only. In DNA, so the DNA molecule which is present from 3' to 5', this strand only act as a template strand, but both not acting as a template strand. The DNA strand which is present from 3' to 5' only, it acts as a template strand. In this template strand, a small portion only transcripted. It occur in 3 steps. So the entire transcription process takes place in 3 steps. In this, the first step is initiation. So RNA polymerase, it binds on promoter of a template strand with the help of sigma factor. Here sigma factor acts as a initiating factor. The second step is elongation. So RNA polymerase enzyme, it synthesis RNA based on the sequence of template DNA strand. In third step, that is the termination. So rho factor, it acts as a terminating factor and stops transcription process, is referred as termination process. So it is a diagram for this question, and in this diagram you should mention the initiation, elongation and termination. Initiation with sigma factor and termination with the rho factor. So it is the diagram for this question. So when you write the answer in this way, definitely you will get full marks for these questions. And remember, from molecular biology, you get 2 5-mark questions.